So next we have Neil, who's an expert in top-down proteomics, as well as many other fields. He and his lab has developed novel mass spectrometry methods and computational strategies for advancing critical areas of biomedical research, such as cancer and systems biology. We very much look forward to his talk and see what advances he has to share with us today.
Thank you, Luke. You're right here. And it's hers is still sharing. Yeah. Good afternoon. Thanks to the organizers for having me come today and share my slides, which I'm doing presently, both with you and those on Zoom. And did I get it? I did. Here we are. Aha, swap displays. Okay, logistics, critical. Yeah, Nikolai, Luke, thank you. And all of the Northeastern team at the Parallel Squared Institute. Very exciting to see more resources coming to our beloved field. I've been watching single cell for some years, 5-6 years, and wondering how we might contribute and get involved. And I'll answer that question today. But one that's on the lips of many is this well-known gap, the genotype to phenotype gap that genomics and genetics have well documented, leading to the doorstep of disease-causing genes, which means proteins in almost all cases. So why hasn't proteomics contributed more to this bridge between genotypes to phenotype, to be better connectors? We've accomplished a great deal, and that's through a lot of resource allocation. So, but when I look at the last 20 years, and particularly the cancer field as a sandbox, to think, well, if you go back 20 years, and many of you may not have been around 20 years ago, that's when I was starting my lab, to precisely date myself, 1999. And, there was a great optimism. There really was, that we're going to detect cancer early. And we're going to do it through proteomics because proteins are close to phenotype, right? If the central dogma holds. Well, now in 2023, we have a lot of multi-omics, a lot of omics platforms to try and connect this bridge. And we're all vying for that. And it's a complicated landscape, to say the least. So wouldn't it be great to level up proteomics and the specificity? And certainly single cell does that, is doing that well. And so how might we level up? Single cell is one way to do that, as reflected on your right-hand side of this slide. And of course, spatial biology and single cell proteomics and single cell omics is born. And, shotgun right out of the gate with a lot of resources and optimism in the last 5 years. Less true is compositional proteomics in terms of the resource allocation, right? So compositional proteomics, still dominated by bottom-up, triptych digestion when using mass spectrometry. Antibody-based platforms are coming along nicely. But of course, either of those platforms give you pieces of what our biology is. Pieces of proteoforms are measured, and usually small pieces. And then you infer the rest. So I'm, of course, here to talk about proteoforms. And for example, the prospect of having, instead of pQTLs, quantitative trait loci, there's all sorts of QTLs out there. And so protein, I would argue that that's peptide QTLs. What about PFRM QTLs or proteoform quantitative trait loci being used to correlate to phenotype, to really be that connector between genotype and phenotype? So those are two important ways to up-level proteomics. And if we do that, we will provide better data, new data types to the whole biomedical research infrastructure of the world. So tell you what, let's table that. I went from 0 to 100 kilometers an hour right out of the gates. Let's go slow it down and go to the regularly scheduled program here, which is proteoforms and single cell proteomics. Can we bring those two things together? By the end of the talk, you'll see that we can. And proteoform tissue imaging, as Nikolai mentioned, this is something the last couple of years we've advanced in multiple ways. And I'll go through that story and the underlying technology that's enabled it. I'll go across some use cases, and I'm fortunate to be part of the HubMap effort, and I'll define what that is for those of you who don't know. But human kidneys and ovaries and ovarian cancer will be part of the story, and then we'll end in the brain. And so what I hope is that the remarks today will help us as a community join forces to not just get single cell proteoform information, which I'll define for those far afield in just a moment, but unlock the Genome Project for proteins to bring the kind of resources to bear, which I think explain a lot of the symptoms of the discussion from this morning's roundtable discussion. So again, decelerating just a bit, the Proteoform was born in 2012 in a conference in Lisbon, Portugal, supported by vendors, I'll add, that, could we get together with protein variants, the inference problem in bottom-up proteomics, isoforms, isozymes, right? There was a need for a term that has since become not just a term, but a movement. And now it's just hit about 1,000 citations in the 10 years since this paper. And so it is a gene-centric term, just like UniProt is a gene-centric database. And they made that transition about this time of 2011 to 2013. And so it means all of the different molecular forms produced from, say, a human gene, but any gene of any organism that creates these proteoforms, which, as depicted here, are those that contain a glycosylation in combination with, say, a phosphorylation that also has a SNP, a biallelic polymorphism that also has an isoform-specific expression pattern. Proteoform is the term that captures all of that variation without ambiguity. A level 1 proteoform is a perfect molecular description of what that molecule is. And that's why it's powerful. And in the last 10 years, joined by those in the Consortium for Top-Down Proteomics, which now is approaching 500 members and almost 200 lab heads, and there's 50 young investigators around the world that are somewhat or largely dedicating their labs to the study and elucidation of proteoform systems biology and proteoform systems medicine. And here, this is partly the reason why. So one of the bridges, if you want to talk about clinical translational proteomics, a bridge between genotype to phenotype, is to get as far as genetics can take you, get dozens, ideally hundreds of patients signed up under IRB approval and do biomarker discovery. And if you do it with proteoform and proteoform resolution, you will be advantaged. We've now shown this in 4 different areas of human biology. The latest is this science paper in 2022, where we did it on 150 liver transplant patients. But really, there's a lot of examples there, and you progress to the right-hand side of this slide, that the concept of a protein or an inferred protein group is not specific enough to connect, to be a best connector for a proteomics measurement. And so providing the decorations without inference is powerful. And I think that future is what I'm here to represent on behalf of hundreds that would like to see major investment in this area and partnership with single cell community. So one place that's become close and is HubMap, so the Human Biomolecular Atlas Program, mapping the human body at single cell resolution. This is hundreds of millions of dollars over 7 years. They spent about $100 million so far. And so the kidney is a good example. Kidney is a good sandbox to say, well, what is the current situation? And that is because KPMP, the Precision Medicine Project run by NIDDK, gave this all a sliding head start. And they're approaching something like 90 cell types in the human kidney and defining this hierarchical all the way down to proteins, which include functional tissue units, so-called FTUs or cellular neighborhoods, and substructures, and of course, single cell. And those cell types largely determined, as was said this morning, by single cell RNA-seq. And the proteomics in this consortium, this large consortium, has been largely multiplexed antibody-based imaging, as also was said this morning and by the last speaker eloquently. And that's the dominant situation. There is proteomics and metabolomics, mass spec imaging in HubMap, but it is not the flagship level platforms that are being used. So, but part of this, and one of the leading program officers at HubMap has this phrase, that I'll share with you. It's, let 1,000 flowers bloom was his approach. Let collaboration happen. And indeed, this story that I'm going to tell you came out of that. I don't know what kind of flower it is, but it's one of the flowers. And so, but there's challenges, right? So how might we do imaging and do it top down and proteoform enabled, get high spatial resolution? Could we get to single cell resolution? And I'll show you the progress along that lines today. But the depth, and the same as bottom up, compositional single cell, we'll have the same sort of challenges. But the throughput in single cell is a major part that I'll address in a few slides. So we've been developing this concept for a while, trying to get ever larger proteoform studies done. And you can see on the far right, that science paper is our latest microcosm of what a whole human proteoform atlas or human proteoform project might look like, including the diagnostic payoff that we could get as a society and as a world. But let me pick up the story of my life at 2018, that the single particle counting there on the lower right of the slide. So individual ion MS, I'll tell you that story briefly, how it underlies proteoform imaging. That's the piece that we'll pick up on. And then the ... on the right, we'll get to before we end. So what we did is took Julia Laskin's technology of nanodesi. This is a liquid junction. There's two capillaries there you see on the left. And I will use my pointer, not just my words. Okay, so here they have two capillaries and there's a liquid junction right here. Then you can change the volume and the size of that, the diameter of that liquid volume. And then that junction slides a few micron a second across the tissue. And literally the tissue was cryosectioned, thin slices, 10, 20 micron thin slices, put on the slide, washed with ethanol, and you do this. There's no antibody labeling. It's a label-free method. But the problem was mass spec imaging at the time was limited to, in spatial resolution, to something like, say, 5, 6 micron. But that wasn't the big limitation for top-down. It was the size of the protein. The size of the protein that you could access, it really was hard to get past 20 kilodaltons, 25 kilodaltons. But this new technology of PIMS, or proteoform imaging mass spectrometry, and Pei Su is in the audience, who came from Julia's lab and joined my lab, and we've had a great time in the past few years. And let me tell you the story of why we can do this up to 80 kilodaltons now, and pushing over 100 will be possible, I'm confident. But a fourfold boost, this opens up the meat of the iceberg in the proteome. And so how this works, so here's the moving liquid junction, scanning across, and I call this mowing the lawn, right? So we mow the lawn back and forth, and an individual snapshot, a few blades of grass are cut and shot into the mass spec. So what you're dealing with is a very small burst of proteoforms coming off, right? There's no digestion, there's no trypsinolysis here. This is direct proteoform extraction from the tissue, and they fly. Okay, how are you going to catch them? How are you going to analyze them? And so what we've got is you'll see this I2MS, or individual ion mass spectrometry. That's the detector, that's the game changer. And then we take a set of these snapshots, all these blades of grass, and we collect them, and then we know what to do with this. We've been doing proteoform analysis for 2 decades, so check that box. And then for any proteoform signal, 2 of which you see in the lower right, we can create an image for that proteoform in this paper, something like 50 micron spatial resolution, and we push that down as I'll show you. So what is I2MS? I know there's really, it's great to have a convergence of cultures and people and countries and different parts of science here. It's fantastic what Nikolai is doing. So let me slow down and give you a sense of what this is, because it really is a major propulsion. It's the most exciting thing that's happened in top-down mass spec in half a decade or maybe even a decade. So here's traditional in the upper left mass spec. Protein complex, there you go. It's a homotetramer, doesn't matter, like 250 kilodalton. So you bring in like 10,000 ions in traditional, what we call ensemble methods, and you do Orbitrap mass spec. So you get this beat pattern in the time domain. here over a few seconds. And that's from the charge states beating frequencies, constructive interference of all the frequencies in the trap from all these ions. And you get isotopic resolution, and that's what these beats mean. Single ion is totally new, totally different. The first thing we had to do was drop the concentration by 1,000-fold, right? So from micromolar to nanomolar, because we're in a single ion regime. And now we can, this spectrum right here is of the same sample. It's just 50 ions. And you can see the alignment of the charge states, right? If you look carefully here, you can see the higher abundance charge states, there's more isotope peaks. That's what those little sticks are single ions of the isotope peaks. And so for any given peak, we can now assign the charge. We know its frequency, therefore we know it's m over z. And that was the breakthrough here, was to that slope of the detection over, say, a half a second to 2-second observation period for 1,000 ions at a time. We can define the slope. This case is a 48 plus. Now we can assign, we know the z, we know the m over z to 1 part in 10 to the 8 precision. Now we can say there's the mass. And it's a total game changer. You no longer require the constructive interference of all these frequencies. And so these are the advantages when you have a single ion, just like single photon imaging, where you know the position of that molecule if it's a single fluor. Here we know the position to 10 to 20-fold greater resolving power, that as a result, the drop in concentration, and then high complexity, no problem. We don't require constructive interference of these ions anymore. Okay, and this was launched commercially by Thermo a few years ago. So this is 4 ions. Yeah, let me know if there's an audio problem. But there's 4 ions there, and you're seeing the slopes created from the integration of signal over a 2-second observation period. And so this might be a 48 plus, this might be a 31 plus, a 30, and a 29 plus, right? So that's the slope that's changing in these plots. And so that was launched a year ago or so as direct mass technology. And so FCOI, I have a conflict of interest with Thermo about that. So back to imaging, okay? We were mowing the lawn, remember? And any given burst of ions, proteoforms that come off the solution. So this is denaturing mode, where we're using acetic and formic acid solutions, some acetonitrile or methanol, and out comes a burst. Any given scan over that 2 seconds, you're going to use this as the detector. And you can see on the right, doing 10 line scans here across human kidney, these are the proteins that we see and proteoforms of them. So naturally, you're going to get the high abundance proteome. You're going to get oxphos. You're going to get primary metabolism, glycolysis, TCA. That's what's represented here with some exceptions. And that's all published. And so the kidney proteoform imaging now, I'll take you through a brief bit of it before we move on. So the glomerulus of the kidney we'll get to in just a minute. That's about a 50 micron structure for reference, okay? So this is a thin slice of a human kidney from HubMap, the consortium, and it's from the cortex to the inner medulla, just a given slice. And here's an example of from the 1,000 or so proteoform signals that we get in this paper, about 50 micron spatial resolution, maybe slightly better. We see different structures, okay? So let me point out, like beyond the mesoscale structures of the cortex and the inner medulla, this vasculature, you can keep your eye on that. Proteoforms of taglin mapped to that here in green. And then here's the several advantages of top-down. There's notable disadvantages, but one of them is, you know, the stoichiometry of the PTMs that you detect. And that's a big difference from bottom-up. And so here is about 50% for heat shock protein beta 1, about 50% of it exists in this phosphorylated form. And I would argue that that stoichiometric measurement of a PTM is a filter for function. And we can talk about that in the Q&A. So zooming into that glomerular structure here in the at left on your right is some of these where you can see the resolution. Okay, we're getting to individual glomeruli. Vimentin is known to be upregulated in glomeruli. So, okay, we're about 40, 50 micron spatial res. You can see it's nothing like multiplexed antibody imaging. There's our vasculature. But we do get the proteoform advantage. For example, this is a biallelic SNP in glutathione S transferase A2. That gene, not the A1 gene, the A2 gene, and that is a difference of a SNP creating a serine or a threonine in two different proteoforms. So that paper took over a year to get through peer review, and we've done a lot since then. And we've been working with Tom Conrads, who saw this technology. He's known to the mass spec community and a big part of the cancer moonshot and the Apollo program. So he sent us, immediately pivoted off of what we were collaborating on, dropped that like a bad habit, moved over to this to do HGSOC, so ovarian cancer proteoforms. He has this huge program in that, a lot of spatial work, a lot of laser capture, microdissection happening with bottom up in that community, along with RNA-seq. So they had defined regions of cancer versus stroma, and we can see proteoform differences like in frill, elevated in stroma, or HMGN2. And now we're pushing, we want to say down to 10 micron, but for here, I'll just say, look, we can get to 20 micron spatial resolution for even abundant or medium abundance proteoforms. So the big question for today, so that was a wind up and a bit of explanation about what's going on. But of course, the big question is, can we push this to single cell, right? We're at 10 micron, 20 micron spatial resolution. Getting that down to 5, mowing the lawn at 5 micron spatial resolution. There are a few labs who have pushed it down to that level, but it's tough. And so I'll address that in a second. But let me just pull out some proteoform biology and out of this, which is the CRIP1 example. If we look at regions of interest that are stroma or tumor enriched here in this volcano plot, some of the top performing ones, let me just focus on CRIP1. So CRIP1 comes in 3 proteoform flavors. It has an unmethylated or mono or a asymmetrically methylated arginine, and they localize differently. So is there biology in proteoform measurement? Yes. And we're just getting to be able to spatially resolve it. So this is a vasculature region up here for the unmethylated form of CRIP, whereas the other methylated forms of CRIP at arginine 67 are enriched in the tumor. So how might we use single ion mass spec to improve single cell proteomics? And that's the big question. And for us, it's a big reveal for today. What we do know how to do, thanks to other groups like Jonathan Sweedler's lab, we can disaggregate cells from a rat hippocampus, which we did, and then put them on slides and control the average distance between each single cell, right? And we can do this on tens of thousands of cells, okay? And then let's just mow the lawn, right? This is a 200 micron droplet, so a pretty big droplet hitting a single cell. And I'll let it go one more time. So there's a single cell here. There it is. And we're going to overcome that with a droplet. It's the lawnmower, right? In my metaphor. Okay, and that's it. And then out of this will come a burst of 10, 20,000 single ions, right? And we know what to do with those. Thanks to the last few years and being focused in the pandemic, I guess, in Chicago, we've been working on this a lot. And so in this burst, you'll see in regions 1, 2, and 3, 2 is where the big droplet overcomes the single cell. And then here you get like a chromatographic peak. You get a peak of proteoform burst, and that subsides over time. Depending on the flow rate, the scan rate, the rastering rate of the droplet, we can control how long that burst lasts, and that's a big area of future optimization. Okay, so now we can do this at scale. This is a readily scalable single-cell proteomics platform, and we just mow the lawn, and this is what we do or what we see from 600 cells, and we call it a cellogram, somewhat like a chromatogram. And that cellogram captures many single cell events, okay? And we can aggregate all of the spectra like we did in tissue. Remember, we were aggregating to create proteoform spectra. We do the same thing here as is on the right of this slide. Many single cell extraction profiles are combined, and there's a lovely proteoform spectrum of histone H1.4, not 1.5 or 1.2 or 1.3, it's histone H1.4 with a selected set of PTMs. So here's some stats for a single PIMS, a single cell PIMS run, that's what we're calling it, single cell proteoform imaging mass spec. And so we covered some real estate. There were 3 different rats, 3 different animals that were used and their cells disaggregated from the hippocampus and put on a set of real estate that we know the average distance. We have a fluorescence map from DAPI staining of what is there. And in sampling what should be around 15,000 or 16,000 cells, we have quality control algorithms in place that we got 10,836 bona fide single cell detection events. I haven't declared the technology that does that, but this processes at about 80 cells per hour. And if you extrapolate forward, there's that's a 20x advance in the throughput of single cell proteomics relative to current platforms. So in our collectors curves, how fast we collect things, this one run was capturing 16 million single ions. Here's the collector's curve as you go across the data set. And this is with charge assignment. Okay, so we don't, just because we detect a single ion doesn't mean we know its charge unambiguously. So there's an attrition there. Then we aggregate these proteoforms, and then this is a conservative estimate, 472 high confidence proteoforms. That is what a standard proteoform processing algorithm, you have to have these isotopic peaks to get an isotopic distribution to have this algorithm detect. We think there's a pushing about 800 or 900 proteoforms there, down to about 0.5% relative abundance. But this is a cool thought as I move quickly through this section, that reassignment is what's on the curve on the far right, on the lower right. So once you've detected these proteoforms and you know they're there, there's a reference library of proteoforms and their ions. Look at the rate at which this accrues. The x-axis here, if it was the same x-axis as here, this would just shoot straight up and over. So our ability to assign known proteoforms is way ahead of our ability to discover them. And here's the aggregated spectrum of this full set that through algorithms, standard algorithms like Thrash in the field, 472 proteoforms were detected and assigned a mass. We were able to identify 170 of those to know their molecular ID, including some PTMs, and they're the abundant, top of the iceberg, so to speak. And there's some Go and the terms right there for the top 3 and their enrichment values in the bottom. Yeah, so this is what this new world of proteoform assignment looks like. It's a single ion from a single cell is the most extreme version of a detection event that you can have. And this one is closed, cell number 8854. That one had 3 ions assigned to the isotopic distribution of that proteoform of aldolase C. Okay, and this PAS or proteoform assignment score, these have to be in register. The mass accuracy here is 1 part per million. So those isotope peaks, it can't be shifted, right? So there is some specificity in here. And it's kind of fun to see, well, even though you might have a low abundance isotope peak in nature, it's single ion world. So you could have detection events that are on the extreme parts of that. But a big abundant one like myelin basic protein, of course, from neurons and astrocytes and microglia cells, or it's going to be abundant. So here's 66 ions in one cell. Okay, so last part, validation to link up with the larger community. Here, if you look up astrocyte in Wikipedia, this is what you see. This is the first image, and it has GFAP and vimentin as markers. That's why it's a yellow star-like looking thing. But GFAP, here's GFAP in our world of single cell PIMS. It is enriched in astrocytes. So of the total of the 10.8 thousand cells that we processed, at least in this data set, we have enrichment and assignment of proteoforms, in this case, astrocytes, for this well-known astrocyte marker, GFAP, N-terminally acetylated. There's the uniprot number. And then here's the reference library. The purple trace here is when we do PIMS, proteoform imaging of the parent tissue. So that's what the purple is. And the black is actually the data set, 10.8K aggregated data set. In 2013, there was a paper that showed S100 beta here as a marker of the soma of astrocytes, whereas GFAP is more on the processes preferentially. And then S100 beta validated as a marker of soma of astrocytes. And then there's our S100B, also N-terminally acetylated, enriched there, and the reference proteoform there as well. So, of course, lots more data, but, this is the theme of the conference and the community, is to give time to converse. But single cell proteoforms, there's 2 or 3 other papers from the top-down proteomics community out there, including from Alexander Ivanov, and there's developers of that drafting right behind the single cell community. And so that should be eagerly awaited. But this technology also, I think it's highly scalable. I think it has a chance to get to 1 million cells pretty fast. And so I hope that from the morning session that we could also use some of the Q&A to talk about this international initiative, and that this technology could bring these 2 communities together, helping give fuel to try and have the resources brought into our field to crush the analytical barriers. This is part of the chicken and the egg issue. When I heard this early morning session we had, I'm hearing symptoms of why we don't have a multi-billion-dollar project to just crush the measurement barriers in our field in proteomics. Genomics has had that, and it was a massive success for the world, and not just in science. If I have a tumor now, I'm getting it sequenced. That's amazing, right? So the consensus, I think, is the big barrier that we need to overcome. And that means writ large, we need enough consensus to, unlock what would be the project to define the human proteome at high compositional resolution. And that we can bring in spatial and single cell resolution as well into this project, which at $1 billion or more, and at, let's say, $1 a proteoform, and we were going to measure 1 billion redundant proteoforms, that's easily $1 billion with a B project. But I don't see any reason not to have an analogy back to the Human Genome Project. And to that goal, this symposium in early October, I think we do need more coordination, because this is where the same single cell conference in Utah is also held. But we can do it over time, if not just in early October. But if you are on the East Coast of the United States or in Europe, why not stop by Chicago on October 3rd and 4th, and then bounce to the start of the single cell conference in Utah this October. So the conclusions, I think proteoforms represent a chance to level up proteomics and would be valuable to everybody. Antibody-based analyses, as was described, and bottom-up community. So there's definitely a lane where we can all work together, because they are proximal to disease phenotypes. That's a different talk, but the evidence is there. And so I think for me personally, top-down has had a challenge to expand and get adoption in the field. But this particular application of just a burst of proteoforms out of single cells, that does take advantage of a core benefit of top-down, no digestion. You just pop it out and there it is, right? So I think getting to like what single-cell RNA-seq has, which has been this explosive growth documented here in the top slides. I won't take you through it. But of course, papers and resources come in that help drop the cost and make it more accessible, right? It's all linked. to getting over 1 million cells there. So I want to thank Sweedler for being Sweedler. And if you know him, you know what I mean. And he's a fantastic source of expertise. And Stas here was the critical element to disaggregate those cells. Thanks for all the funding. Otherwise, it wouldn't have happened. And hopefully we can pave a road to the future. Thank you.
Thank you so much for the great talk. Really exciting. It seems to be like a great convergence of different technologies at this conference, as we've seen from our last several speakers and others today. Yeah, thank you so much. I just want to open it up to the floor for any questions that we might have.
Really great talk, and the whole nanodesi is like, yeah.
Right. It was right there.
Beautiful. So can you, I assume you're using a different solvent, different extraction solvent when you are pulling out intact proteins instead of, small molecules. And have you can you tune the solvent? I could have just talked to this guy, right?
You can. Hey, you want to take it? Have Pei answer.
Okay.
Yeah, absolutely. So what is your favorite?
Short answer is yes.
Yes. I guess I'm just wondering how different your detection is when you change your extraction solvent.
So we typically do denatured proteoform extraction from cells, and that has a pretty high organic content. So that is also to the benefit of ionization, right, out of nanodesi.
Doesn't if you're extracting small molecules, that's also, when Julia was at PNNL, it was like 90% methanol.
Yeah, you had a front row seat to the early stages of this technology. I see. I think it's the ethanol wash, because is it a delipidation? Would you agree that a high content ethanol? Actually, we do 3 washes.
Okay,
so I think
you're getting rid of that before you come in.
I think that's why you're having this lipid moment.
Right. Okay, thank you.
Yeah.
Just a great talk. Just to follow up on Ryan's question. So you deposit the cells on a glass slide or a platform. You just drop them dead there. You freeze them. You fix them with ethanol. What is going on there?
Some oxidation is going on to my dismay. But
you deposit them dry. You fix them. You put them on the.
No, not fixed. They adhere. So Stas is been doing this a long time, and Sweedler has come up with a single cell metabolomics. It was chosen in Nature as like one of the 6 technologies to watch for the next year, and this is how he does it. So we didn't contribute anything there other than knowing Stas and Jonathan. So the bigger the cell, the more it adheres. You can kind of wash them off if you're not gentle with it, but that's it. They stick.
All right, thanks.
Yeah, these are just IDO glass slides. That's it.
I have a just quick question regarding cell size. It looks like this is a very sensitive method, and you've analyzed some brain cells. Have you analyzed, say, smaller cells, and what do you see in the distributions of proteoforms for different cell sizes of different tissues, for example? Or
not a systematic study, Luke. I love your question. Very precision question. And if I get to this, so there's some microglia, and those are little small ones. And these are the ones Stas was worried about washing away, to Professor Ivanov's point. But to your point, microglia are smaller, and we were able to assign 700 or so of them. And as long as they're on the glass slide, we're gonna sample them. And I bet. Let's go do this, Pei, tonight, please. If you can have the result back, that would be great. Look at the number of single ion detection events in these 3 cell types. I bet, because what I learned at this conference is that, the smaller the cell, the fewer the protein groups. And so we'll have that for you tomorrow, Luke.
I appreciate it.
Okay.
There's a snapshot.
Oh,
It's a snapshot that's a 600 cell cellogram. You see up and downs, and that is your ion count for different cells. But we don't have specific numbers, but that is an indicator.
Do you?
Yeah, the next one. Yeah.
oh,
Interesting.
yeah, there you go. It's just like a chromatogram to view it. Yeah.
So through this kind of mowing the lawn technique, can you also maybe take a snapshot and estimate cell diameter before you do it? And
Yeah,
on
we
to
have we have images of a large number of cells. So we, just like in that movie, we could get that as well. That's an interesting way to combine the optical part of this with the single cell PIMS.
thank you.
I have a question about cell isolation, Neil. Do you think that you might be able to use expansion microscopy to preserve the spatial location of single cells rather than put them on a glass slide and then do your magic?
That is a you want to write a grant about it? That's a great idea. And the single cell RNA community going spatial. You heard from Erwin earlier about how he's dancing on that next step. So we've been thinking about this for fewer numbers of years than others have, but that sounds like an awesome. That seems completely registerable. You could put them in register and extract that info. We do also hope to get out directly out of tissue, get down to 5 micron or less, and we haven't given up that program either.
And another quick follow-up question on the main benefits of the droplet that gets on top of the single cell. Why do you add that liquid on top prior to ionization? Yeah, exactly this one.
Well, it's inherent to the liquid junction, and we can change the size of the liquid junction. We could save change the solvent composition. I think that's a big area. We know we're not extracting the majority of it out, the proteins out. So I think there's a big move here to increase the number, but you got to balance that with throughput. So there's a balance there. But your point was different. I think your point was there a way to do it differently? You may be thinking of a different geometry than the default one here, where the droplet has to go over the cell, and that's what's being rastered. So maybe I'm not gathering your.
That's okay. We can discuss this later.
Yeah.
Thank you.
Yeah.
Hello, that was a wonderful talk. Actually, I'm a little jealous, like DESI, single cells, single charge detection, plus, well, actually detection. Apparently a good orbit trap.
To that point, I know that's a serious statement. I've been feeling that for years. What I propose is that we unlock a major increase in support for funding, and then we would all benefit. So there's no requirement for jealousy.
No, I totally get it. That's actually a great proposal. I'm curious. So this 80 cells per hour, and you show us 700 plus 1500 plus, so 2800 cells, the 35 hours of instrument time. So apparently there is somewhere a bottleneck, because otherwise you would probably do more than that. So where is the bottleneck?
Yes. Oh, and you want to know what it is?
Yes.
Yeah. So it's the 40 micron a second scan rate, right? I mean, or you maybe, and this is what Nikolai was hinting at, change the distance between the cells, right? Because there's some real estate in between each one, and you close down the distance, the cell-to-cell distance, and you speed up, you can go faster. And we're extracting each peak on the cellogram is 6 seconds wide. So that's the inherent. I think that's a great hint at how we can speed this up. Smaller droplet, less distance cell to cell. You could probably get 2, 3. I want to say 4 or 5x, but I'll start it with 3x increase in these throughput numbers.
Thank you.
All right.
Luke, could I just take the extra 30 seconds and
Yes.
and this thing that came up, and it was hit upon by the panel, and something we'll talk about in October in Chicago. But this synergy with next-gen proteomics, single molecule protein sequencing, single cell analysis, I see it as exactly that. And the private sector has taken more risk and put in more direct resources into that than federal funding of science. And I think that that's a situation that we can try to point out that that's true. But you have these companies like, and I'm excited to hear Bryan from Quantum Sci and Nautilus, and there's a variety of proteomics companies. A lot of them went public. A lot of them are heavily capitalized. They've been financed. And so I think this is the chance. I see an opportunity here, and we just need to get the government on board. So thanks for that, Luke.
Thank you very much.